Hello guys, I welcome you all to this CSIR NET platform of Unacademy, and we are doing cell signaling part 2. Myself Himanshu Yadav. So I welcome you all to this session on cell signaling which we will be continuing today. हम लोग ने ये सेशन कल स्टार्ट किया था और हम लोग इस सेशन को आज फिर से कंटिन्यू करने वाले हैं, जिसमें हम लोग आगे का प्रोसेस हम लोग स्टडी करेंगे. So I welcome everyone. I hope everyone is there online. So hello to everyone. So before we start, I would like to tell that you need to go to the Google Play Store, and by going to the Google Play Store you need to go and install the Unacademy's learning app. And by installing the Unacademy learning app, you need to go and you need to select your goal. In the select your goal category, you need to select CSIR UGC NET. So once you do that, you will be getting access to various courses from top educators in this particular category which are available for free. If you want to go for more, अगर आप लोग ज़्यादा के लिए जाना चाहते हैं, then you need to go for plus subscription, because plus subscription में काफ़ी advantages हैं. जैसा कि आपको मैं बताना चाहूँगा कि in the plus subscription you will be getting access to multiple courses in a single subscription. You will be having a doubt session, and you will be getting a PDF. You will get direct interaction with the educator. You will solve the previous year paper questions. You will be solving expected questions and quizzes, and then you will be also getting homework questions. So there is a lot of benefit in plus. And plus ka subscription you can get it for 1 month, 3 months, 6 months, 12 months and 24 months. But I would recommend that each one of you go for 12 months to 24 months of subscription, because this is an ideal time that is required for completing the syllabus, because as you all know, CSIR ka syllabus is very vast. So if you go for 12 months to 24 months of subscription, then it is possible to finish off the course in a required amount of time. And you can enter my referral code HY10 for getting 10% off. So once you type the referral code, then you need to apply it, and you can move to the payment, where you can do the payment either by the debit card or credit card or the EMI, and these EMI are all no cost EMI. You can also do the payment by the net banking. And इसके साथ ही साथ this is for the plus classes. इसके साथ ही साथ मैं आप लोग को ये बताना चाहूँगा कि YouTube पे हमारे सेशंस चल रहे हैं, और आने वाले समय में हम लोग ने आज से ही हम लोग ने लक्ष्य JRF 2021 स्टार्ट किया है, जहाँ पे लाइफ साइंसेज में हम लाइफ साइंसेज बाय कुमकुम मैम एंड बाय हिमांशु सर कराया जाएगा. साथ ही साथ you will be getting maths by Amit sir and Rahul sir, and chemical sciences by Richa ma'am. So आज इसका opening session हुआ है. You can go and visit the session online. So this was the opening session for Lux JRF 2021. So Kumkum ma'am and myself will be conducting the life sciences classes, where you will be getting access to the conceptual where you will be getting the time to develop your conceptual understanding as well as enhancing your analytical ability. So let's start the session. हम लोग सेशन को स्टार्ट करते हैं. So कल हम लोग ने सेल सिग्नलिंग में काफ़ी पढ़ लिया था. सो यहाँ पर हम लोग अब देखने जा रहे हैं कि सेलुलर सिग्नलिंग इट अफेक्ट्स द चेंज इन द शेप. सो सिग्नलिंग का कैसा इफेक्ट होता है शेप चेंज पे. सो सिग्नलिंग पाथवे कैन ऑल्सो इफेक्ट द ओवरऑल बिहेवियर ऑफ अ सेल. सो फॉर एग्जाम्पल, देर इज चेंजेस इन द सेल शेप. सो चेंजेस हो जाते हैं सेल शेप में. तो जैसा कि आपने देखा कि यहाँ पर Just a minute. तो जैसा कि आपने देखा कि यहाँ पर मेटिंग फैक्टर एक्टिवेट रिसेप्टर करता है, एक्टिवेट्स करता है रिसेप्टर को। और ये एक GPCR का रिसेप्टर होता है जहाँ पे इनिशियली प्रोटीन GDP से बाउंड होती है, बट जैसे ही लिगेंड मॉलिक्यूल रिसेप्टर से बाइंड करता है, देन उस केस में G प्रोटीन विल बाइंड टू द GTP एंड इट विल गेट एक्टिवेटेड। इस केस में वो फॉस्फोराइलेशन कैस्केड एक स्टार्ट करती है जहाँ पे FUS3 प्रोटीन का फॉस्फोराइलेशन होता है, और उसके बाद इट मूव्स टू द प्लाज्मा मेम्ब्रेन जहाँ पे वो फॉर्मिन का फॉस्फोराइलेशन करके एक्टिव फॉर्मिंग में कन्वर्ट करती है। एंड फॉर्मिन इनिशिएट्स करता है ग्रोथ ऑफ माइक्रोफिलामेंट्स दैट फॉर्म द SHMOO प्रोजेक्शन। सो फॉर्मिन इज the protein which is important for the assembling of the actin subunits. So इसका phosphorylation से इसका एक्टिवेशन होता है। सो यहाँ पे आपने देखा कि सेल सिग्नलिंग में कैसे सेल शेप के लिए सेल काम कर रहा है। Then there are some fine tuning of the response. So there are 4 aspects of fine tuning to consider. So first there is amplification of the signal, then there is specificity of the response, then there is overall efficiency of the response which is enhanced by scaffolding proteins, and at the last there is termination of the signal. So there are 4 aspects of fine tuning that we need to consider. So ये 4 aspects बहुत important हैं because it leads to the fine tuning of the response. So first there is amplification of the signal, then there is specificity of the response, then there is overall efficiency of the response which is enhanced by scaffolding proteins, and then there is termination of the signal. So let's move forward. और सिग्नल का एम्पलीफिकेशन में क्या होता है कि देर इज एंजाइम कैस्केड्स विच एम्पलीफाई द सेल्स रिस्पॉन्स. सो at each step the number of activated products is much greater than in the preceding step. So enzyme का cascade it amplifies the cell's response. Specificity of cell signaling and coordination of the response. So different kinds of cells have different collection of proteins. So different kinds of cells have different collections of proteins. So these different proteins they allow cells to detect and respond to different signals. So even the same signal can have different effect in the cells with the different proteins and pathways. So pathway branching and crosstalk further helps the cell to coordinate the incoming signals. So there is a signal molecule which will come and it is attached to the receptor molecule, and it will in turn activate the relay molecules which will lead to response 1. In case of cell B, the pathway it branches leading to two responses. There is one response as 2, another response as 3. Then in case of cell C, there is a cross talk between two pathways. So एक pathway चल रहा होगा और उसमें दूसरा pathway आ जाएगा. So ये this is the second pathway, this one is the first pathway. So यहाँ पर एक particular pathway में जब cell का activation होगा, so वो activation या inhibition कराएगा दूसरे जो पाथवा चल रहा होगा उसमें कैस्केड का. सो यहाँ पे क्रॉस टॉक हो रही है एंड देन देयर इज अ डिफरेंट रिसेप्टर दैट लीड्स टू अ डिफरेंट रिस्पॉन्स. सो एक पर्टिकुलर टाइप के रिसेप्टर से एक पर्टिकुलर टाइप का रिस्पॉन्स आता है और दूसरे टाइप के रिसेप्टर से दूसरे टाइप का रिस्पॉन्स आता है. सो यहाँ पे सेल डी है जो कि डिफरेंट टाइप ऑफ रिसेप्टर, इट लीड्स टू अ डिफरेंट रिस्पॉन्स. So this is the case where the signaling molecule it leads to the activation of the relay molecule and it leads to the response 1. Then there is response 2 and response 3 where cell B, in case of cell B, the pathway branches leading to two responses. This one is the cell C जहाँ पे cross talk हो रही है between two pathways, and then there is cell D जहाँ पे different receptor leads to a different response. So next we are going to study about the signaling efficiency. So scaffolding proteins and signaling complexes they are important because scaffolding proteins are large relay proteins to which other relay proteins are attached. So scaffolding proteins they can increase the signal transduction efficiency by grouping together different proteins involved in the same pathway. So in some cases scaffolding proteins may also help to activate some of the relay protein. So in some cases the scaffolding proteins may also help to activate some of the relay proteins. और scaffolding protein का advantage ये होता है कि they increase the signal transduction efficiency by grouping together different proteins that are involved in the same pathway. So this is an example of the scaffolding protein जो कि attached है 3 different protein kinases से, and as soon as the signaling molecule comes and attaches to the receptor, then it goes and attaches to the receptor. Scaffolding protein it attaches to the receptor. Then there is termination of signal. So signal का termination होना भी उतना ही important है. There are some inactivation mechanisms that are essential aspect of cell signaling. So if a ligand concentration falls, so fewer receptor will bound, and unbound receptor revert to an inactive state. So apoptosis, we are now going to study about a little bit about apoptosis. So apoptosis integrates multiple cell signaling pathways. So apoptosis is programmed or controlled suicide. So components of the cells are chopped up and packaged into vesicles that are digested by the scavenger cells. So apoptosis prevents enzymes from leaking out of a dying cell and damaging the neighboring cell. So apoptosis is also a program cell death, where the cells are chopped up and packaged into the vesicles that are digested by the scavenger cells. So apoptosis prevents enzyme from leaking out of a dying cell and damaging neighboring cells. So apoptosis was studied in the soil worm C. elegans. So apoptosis is important in shaping an organism during embryonic development. So the role of apoptosis in embryonic development was studied in C. elegans. So in C. elegans, apoptosis results when the proteins that accelerate apoptosis override then those that put the brakes on apoptosis. So C. elegans में apoptosis तब होता है कि जो प्रोटीन्स एक्सेलरेट करती हैं apoptosis वो override कर जाती हैं, they override the cells that put brakes on the apoptosis. So in that condition there is apoptosis in C. elegans, and this is an important concept. This has been asked in the examination several times. So there is initially what happens is when there is no ligand molecule and the receptor for death signaling is empty. So in that case, CAD9 protein it inhibits the CAD4 activity, and CAD4 initially it also leads to the inactivation of CAD3 gene. So this CAD4 and CAD3 are not activated, and initially there is no cell signaling that is leading to the apoptosis. But when the death signaling molecule arrives, as in the second case, so it leads to the activation of the protein which activates CAD4, and eventually CAD3 will be activated, and it will lead to the production of other proteases and nucleases. So cell forms blebs, and nuclear disintegration also starts to take place, and thereby this is the death signal, so apoptosis will start. So this is a classic case where the CAD9 binary switch is activating, and it is also in the deactivating stage. So it will lead to the death signal when the receptor molecule is when the ligand molecule is present on the receptor. So this is the CAD9 protein which inhibits CAD4 activity, and there is a receptor for death signaling molecule. So CAD4 and CAD3 are activated. Initially they are not activated initially because there is no death signal, or the ligand molecule is not there on the receptor. So active CAD9 protein, it inhibits the CED activity. CD4 and CD3 are inactive proteins. Then as soon as the death signaling molecule comes, it activates the it leads to the inactivation of the CAD9 protein, and CAD9 protein का जब inactivation होता है तो it will lead to the activation of CAD4 and CAD3 proteins, so which lead to the activation cascade, and other proteases and nucleases are synthesized, and it leads to cell forming blebs, and nuclear disintegration takes place. So apoptosis हो जाती है. So there are apoptotic pathways and signals that trigger them. So caspases are the main proteases that carry out apoptosis. So caspase are the important concept. So apoptosis can be triggered by an extracellular death signaling ligand. So it can be triggered by DNA damage in the nucleus, or if there is protein misfolding in the endoplasmic reticulum, then also apoptosis can be triggered. So ये तीन condition है where apoptosis can be triggered. So it can be triggered by an extracellular death signaling ligand. It can be triggered by DNA damage in the nucleus. It can be triggered by protein misfolding in the endoplasmic reticulum. So if you have any doubts, if you have any queries, then you can post it in the comment box. I hope आप लोग को समझ में आ रहा होगा, and if there is no doubt, and then let's proceed forward. So apoptosis is evolved in the early in the animal evolution, and it is essential for the development and maintenance of all cells. So apoptosis, it evolved early in the animal evolution and is essential for the development and maintenance of all cells. So apoptosis may be involved in some diseases. For example, in case of Parkinson and Alzheimer's, the interference with apoptosis may contribute to some cancers. So interference with apoptosis may contribute to some cancers. So this is an important part. तो जो हमारे डिजिट्स जो होती हैं, इंटरडिजिटल टिश्यू होती है, इसका पर अपॉप्टोसिस होता है, एंड दैट इज़ व्हेन द स्पेस बिटवीन द डिजिट्स इज कम्स इनटू प्ले. सो जो सेल्स होती हैं वो अपॉप्टोसिस हो जाता है उनका, और इंटरडिजिटल टिश्यू का अपॉप्टोसिस होने के बाद देयर इज स्पेसिंग बिटवीन द डिजिट्स. सो दिस इज द इंटरडिजिटल टिश्यू. तो यहाँ पे अपॉप्टोसिस होता है, और यहाँ पे अपॉप्टोसिस होने के बाद देयर इज द फॉर्मेशन ऑफ डिजिट्स. सो देयर इज स्पेस बिटवीन द डिजिट्स. So there is a signal molecule which will act on the receptor form, so it will lead to the transduction part in which the relay molecules will be there, and which will lead to the activation of the cellular response. So cell signaling, it consists of basically three pathways. First is the three parts. First is the reception, second one is the transduction, and third one is the response. एंड रिस्पॉन्स होगा जहाँ पे एक्टिवेशन ऑफ सेलुलर रिस्पॉन्स हो जाता है. नेक्स्ट वी आर गोइंग टू स्टडी सम ऑफ द इम्पॉर्टेंट सेल सिग्नलिंग. सो दिस इज एन एग्जांपल ऑफ विंट बीटा कैटेनिन सिग्नलिंग. सो ये एक इम्पॉर्टेंट कंसेप्ट है स्टडी करने के लिए. तो होता क्या है कि जब विंट का लिगेंड एब्सेंट होता है तो फ्रिजल्ड जो प्रोटीन होती है वो इनएक्टिव स्टेट में प्रेजेंट होती है. फ्रिजल्ड रिसेप्टर जो है वो इनएक्टिव होता है, और LRP5-6 भी इनएक्टिव स्टेट में रहता है. उसका फॉस्फोराइलेशन नहीं हो पाता है, जिसकी वजह से क्या होता है कि बीटा कैटेनिन जो प्रोटीन्स होती हैं उनका प्रोटियोसोमल डिग्रेडेशन हो जाता है, क्योंकि एक्सिन और GSK3 बीटा एक्टिवेट हो जाती हैं, और एक्सिन प्रोटीन इज अटैच्ड टू APC एंड CK1. सो देयर इज अ कॉम्प्लेक्स ऑफ APC, CK1, GSK3 एंड एक्सिन which leads to the phosphorylation of the beta catenin, and beta catenin is degraded when there is no Wnt ligand. So जब Wnt का ligand present नहीं होगा, याद करने वाली बात यह है, Wnt जब ऑफ स्टेट में होगा, जब Wnt का सिग्नल नहीं प्रेजेंट होगा, जब वो ऑफ स्टेट में होगा, then there will be proteosomal degradation of beta catenin and the transcription will be stopped. so no further proteins will be synthesized in that case. But जब wind का signal आता है and it comes and attaches to the Frizzled receptor, so it leads to the phosphorylation of LRP56, so which eventually leads to the phosphorylation of DVL. so उसकी वजह से क्या होता है, उनका जो एक्सिन APC CK1 GSK3 कॉम्प्लेक्स है उनका डिस्ट्रक्शन हो जाता है एंड बीटा कैटेनिन इज फ्री. सो बीटा कैटेनिन का अक्यूमुलेशन हो जाता है एंड इट लीड्स टू द ट्रांसलोकेशन इनसाइड द न्यूक्लियस where it leads to the activation of the TCF, it leads to the transcription of TCF and LEF related gene products in the case where axin and FGF and BMP and SOX are synthesized. So when the wind signal is present on the frizzled receptor, then beta catenin will be accumulated and it will lead to the transcription of the mRNA of the DNA. And when the wind is absent, it will lead to the degradation of the beta catenin protein. So ये सेम क्वेश्चन कई बार पूछा गया है एंड वहाँ पे कंफ्यूजन हो जाता है एग्जामिनेशन हॉल में. सो इसको अच्छे से समझ लीजिए at how it is taking place. So if you have any doubts or if you have any queries, so you can post it in the comment box. नेक्स्ट वी कम टू जैक स्टार्ट सिग्नलिंग. सो ये भी एक इम्पॉर्टेंट टाइप की सिग्नलिंग है. सो जैक स्टार्ट सिग्नलिंग में क्या होता है कि देर इज़ जैक एंड देर इज़ स्टार्ट. सो इट इज़ अटैच टू द रिसेप्टर इनिशियली एंड वहाँ पे साइटोकाइन आएगा. सो साइटोकाइन आर द लिगेंड्स इन दिस केस. सो जैसे ही साइटोकाइन आके रिसेप्टर पे अटैच होता है तो इट लीड्स टू द फॉस्फोराइलेशन ऑफ द स्टार्ट प्रोटीन्स. And start proteins then they move inside the nucleus where they lead to the transcription of the DNA and it will lead to the synthesis of there is aberrant gene expression where HP1 alpha and things like that will be synthesized, and then there will be arginine methylation also. So arginine methylation will take place on H4 and H2A. So JAK2 controls the arginine methylation pathway and JAK2 also controls the H3Y41 this pathway. So cytokine receptor होता है JAK STAT signaling में. So ये एक important concept है remember करने के लिए. So this is the example of the apoptosis signal that is taking place. So there is a fast ligand. So fast ligand is a kind of a death ligand. So it will come and attach to the receptor molecule, which are fast cell surface receptor molecule, which are clustered, and it will lead to the activation of the adapter protein, which will make the inactive caspase 8 into active caspase 8, एंड जो एक्टिव कैस्पेस 8 होगा उससे इनएक्टिव कैस्पेस 3 को एक्टिव कैस्पेस 3 में कन्वर्जन हो जाता है, जिसकी वजह से अपोप्टोसिस इनिशिएट हो जाता है. So it will lead to the apoptosis initiation. So ये एक important concept है. This concept has been asked several times in the exam. So then there are some general principles of signal transaction. So signal transaction, it refers to the overall process of converting the extracellular signals into the intracellular responses. So in signal key players जो होते हैं in signal transaction में, they are the signaling molecules, they are the receptors, they are the signal transaction protein, and then there are the secondary messenger and effector proteins. So cells respond करते हैं to signal by changing the activity of the existing enzymes and the levels of expression of enzymes and cell components by gene regulation. So receptors and signal transaction systems have evolved to detect and respond to hormones, growth factors, neurotransmitters and pheromones. So also to oxygen, nutrient, light and touch. So there is an enormous number of signal molecules and receptors in the cell. So in contrast, there are relatively few types of intracellular signal transaction systems. So ये एक आइडियल प्रोसेस था जहाँ पे सेल की सिग्नलिंग हो रही थी. This is the ligand molecule that is coming from one cell, and it will go and bind to the receptor protein on the cell membrane of the responding cell. Initially the cell receptor was inactive, but when the ligand will bind, it will lead to the activation of the cell surface receptor, which will lead to the activation of the signal transaction and secondary proteins, which will lead to the maturation of effector protein. So then the effector protein will move inside the nucleus, and it will lead to the modification of the gene expression and development. So signaling कई तरह की होती है. Then there is endocrine signaling. Endocrine signaling जो कि hormones के केस में होती है, that in that case the hormones are secreted into the blood by the endocrine glands. Then there is paracrine signaling, जहाँ पे एक secretory cell होता है और adjacent cell होता है उसके बगल में. So secretory cell leads to the activation of the adjacent cell. So this is an example of paracrine signaling. Then there is autocrine signaling, जहाँ पे सेल अपने ऊपर खुद ही टारगेट साइट्स बना के उसमें ligand molecules को पहुँचाता है, जिससे कि एक एक्स्ट्रा सेलुलर सिग्नल का आता है. एंड देन देयर इज सिग्नलिंग बाय प्लाज्मा मेम्ब्रेन अटैच्ड प्रोटीन्स. सो दिस इज ऑल द डिफरेंट टाइप्स ऑफ सेल सिग्नलिंग. सो एंडोक्राइन सिग्नलिंग, इट एक्ट्स ओवर लॉन्ग डिस्टेंसेस विद इन द ऑर्गेनिज्म. पैराक्राइन signaling acts over very short distances, for example between the neighboring cell. So neurotransmitters and developmental signals typically act in this manner. So in the case of autocrine signaling, the cells release the ligand that bind to their own surface receptor, modulating the activity. So many growth factors act in this manner. Finally, the signaling systems involve plasma membrane attached proteins. They act via direct cell to cell contact. Then there are receptors. So cell surface receptor, they bind to the ligand via their extracellular domains. So in all cases, the binding causes a conformational change in the receptor that leads to the transmission of an intracellular signal. So binding specificity and affinity are determined by the extent of molecular complementarity between the ligand and the receptor. So a given receptor may exhibit specificity for a ligand and or a closely group related ligands. So a given ligand may bind to a number of different types of receptors that exhibit different effector specificity. Further, two receptors that bind different ligands may signal via the same intracellular signal transduction system even within a single cell. So हम लोग ने कल भी स्टडी किया था about kinases and phosphatases. That kinase that brings about the phosphorylation of the amino acid side chains in the target protein, whereas phosphatases they hydrolyze the phosphate of these residues. So then there are GTPase switches which act so GTPase switch protein also play an important role in the intracellular signaling transduction. So GTPase are active when bound to the GTP and inactive when bound to the GDP. So the time frame of activation depends on the GTPase activity of these proteins. So proteins known as guanine nucleotide exchange factors, they promote the exchange of GTP for GDP and activate the GTPase. So protein known as GTPase activating protein, they stimulate the rate of GTPase hydrolysis to GDP and activate the GTPase. So we will cover two classes of GTPase switch protein, that is trimeric G protein and then there is monomeric G protein. So trimeric G protein, it interacts directly with the receptor, whereas small G protein interact with the receptor via adapter proteins and guanyl exchange factors. Then there are signaling secondary messengers like cyclic AMP, then there is 1,2-diacylglycerol that is known as DAG, then there is inositol 1,4,5 triphosphate that is known as IP3, and then there are calcium and phosphoinositides. So the function of cyclic AMP, GMP, DAG and IP3 are summarized in the figure. So secondary molecules are there. They are the small molecules that diffuse rapidly through the cytoplasm to their protein targets. So another advantage of secondary messenger is that they facilitate the amplification of the extracellular signal. So this is the signal amplification in signaling pathways. Then there is a reversible, there is ligand binding and receptor activation. So there is reversible kinetic equation for the ligand binding to the receptor. So ligand will be ligand plus receptor will be forming a ligand receptor complex. So dissociation constant for this reaction will be R into L upon RL. So where L is very similar to KD, so the receptor is 50% saturated in that case. So when the ligand concentration will be 10 kD, the receptor is 90% saturated. So at ligand concentration 0.1% kD, the receptor is 10% saturated. So typically the kD for ligand binding is higher than the basal concentration of the ligand. So this is needed for cells to optimally respond to the changing ligand concentration. So interestingly, the level of physiological response typically does not strict parallel binding. So the number of receptors in a cell is very important in setting the physiological response. So a decrease in the receptor number reduces the response and vice versa. So this is a graph between concentration of the ligand and fraction of maximal binding of the cellular responses. So fraction of maximal binding of cellular or cellular responses, it is present on the y-axis, and there is relative concentration of the ligand that is present in the x-axis. Then there are structures of GPCR. So GPCR are most numerous class of receptor in most eukaryotes. So receptor activation by ligand binding activates an associated trimeric G protein, which in turn interacts with the downstream signal transduction protein. So all GPCRs are integral membrane proteins that have a common 7 transmembrane structure. ये हमने देखा था. So the ligand hormone binding domain is formed by the amino acid that are located on the exterior side of the membrane. So likewise in rhodopsin, its light absorbing chromophore 11 cis retinal is located within the transmembrane segment interior of the protein. So GPCR, they interact with the G protein via the amino acid in the C3 and the C4 cytoplasmic regions. So these are the C3 and the C4 cytoplasmic regions where the G protein interact. So this is the case of the G protein coupled receptor cell signaling. So initially क्या होगा कि the hormone is present and then there is exterior side and then there is cytosolic side and the receptor is an inactive state. In that case, the GDP protein is bound to GDP and it is and there is inactive effector molecule. So there is a trimeric G protein that is having alpha, beta and gamma subunit. So what happens when the hormone molecule comes and binding of the hormone, it induces a conformational change in the receptor. So it will lead to the activation of the receptor. So activated receptor, it binds to the G alpha subunit. So once it binds to the G alpha subunit, then there is a conformational change in the G alpha subunit and triggering the dissociation of the GDP. So G alpha subunit it gets separated from the G beta gamma subunit and it is GDP linked now because it will lead to the activation of the effector protein. So there is an so G alpha protein, it leads to the activation of the active effector protein, and then there is hydrolysis of GTP to again GDP, which causes G alpha to dissociate from the effector and reassociate with the G beta gamma. So there is hydrolysis of GTP to GDP which causes G alpha to dissociate from the effector and reassociate with beta gamma. So GPCR वहाँ पे भी प्रेजेंट होते हैं वेयर दे रेगुलेट द आयन चैनल्स. सो देर इज म्यूसिकार्निक एसिटिल कोलाइन रिसेप्टर. सो जो न्यूरोट्रांसमीटर एसिटिल कोलिन होता है, इट बाइंड्स टू 2 रिसेप्टर्स नोन एज निकोटिनिक एंड म्यूसिकार्निक carinic acetylcholine receptors. So nicotinic receptor, it is itself a ligand gated ion channel that opens on ACH binding. So this receptor is located in the neuromuscular junction of the striated muscles. So the muscarinic acetylcholine receptor is a GPCR found in the cardiac muscle cell and that is coupled to inhibitory G protein. So the binding of the acetylcholine to this receptor triggers the dissociation of G alpha GTP from the G beta gamma, which in this case directly binds to and opens the potassium channel. So the movement of potassium down its concentration gradient to the outside of the channel increases the positive charge outside the membrane, hyperpolarizing the cell. So this results in the slowing of the heart rate. So यहाँ पे heart rate में भी control है G protein का. So हम लोग देख सकते हैं किस तरीके से. So GPCR में यहाँ पर भी they regulate the ion channels in the case of rhodopsin as well. So rod cells are important as they capture in the low intensity light having a broad range of wavelength. So there are closely related color pigment receptors that respond to more limited regions of the visual spectrum. So यहाँ पे 11 cis retinal convert हो जाता है all trans retinal में by the G protein family, and that is where it leads to the activation of the opsin protein. this is some details about. Then there is synthesis of cyclic AMP, and cyclic AMP is broken down to AMP by cyclic AMP phosphodiesterase. So diesterase is the enzyme. So GPCR, they regulate the adenyl cyclase, and adenyl cyclase they activate the other effector proteins. So adenyl cyclase is an effector enzyme that synthesizes cyclic AMP. So a given cell type can express multiple type of GPCR that all couple to the adenyl cyclase. So the net activity of adenyl cyclase thus depends on the combined level of the G protein signaling via the multiple GPCR. So in liver, the GPCRs for epinephrine and glucagon both activate the cyclase, and in case of the adipose tissue, the epinephrine, glucagon and ACTH activates the cyclase via G-alpha stimulatory GTP, while PGE1 and adenosine inactivate the cyclase via G-alpha 1 GTP. So यहाँ पे activation of gene transcription है by GPCR signaling. So GPCR, they regulate the gene transcription by cyclic AMP and PKA signaling. So जैसे ही G protein coupled receptor में ligand आके bind करेगा, it will lead to the activation of adenylyl cyclase and it will form the cyclic AMP. Now this cyclic AMP will go and bound to the regulatory subunits of PKA, and it will phosphorylate the regulatory subunits, so the catalytic subunits will separate, and once the catalytic subunit will separate, they will move inside the nucleus, where they will be phosphorylated by the ATP by the Krebs protein. So it is CRE binding protein, and it which bind to CRE sequences that are upstream of CMP regulated genes. So only phosphorylated Krebs has DNA binding activity. So Krebs interact करता है with other transcription factor to enable the RNA polymerase II machinery at these promoters. So in liver, the glucagon signaling by this pathway activates the transcription of gene that is needed for gluconeogenesis. So आप देख रहे हैं कि G protein coupled receptor का कहाँ कहाँ पे part है. So then GPCR, they activate phospholipase C also. Then there is IP3 DAG that elevates the level of calcium ion, जो कि हम लोग ने कल देखा था. Then there is nitric oxide signaling. So there is cyclic GMP signaling in case of nitric oxide. So a related signaling pathway involves phospholipase C that operates in the vascular endothelial cells and causes adjacent smooth muscle cells to relax in the response to circulating acetylcholine. So in case of nitric oxide cyclic GMP signaling pathway, the downstream target of the calcium calmodulin is the nitric oxide synthase, which synthesizes the gas nitric oxide from arginine. So nitric oxide, it diffuses into the smooth muscle cells and causes relaxation by activating guanylyl cyclase and increasing cyclic GMP. So as a result, arteries in the tissues such as the heart, it dilates and increases the blood supply to the tissues. So nitric oxide is produced from the drug nitroglycerin, which is given to the heart attack patient and patients being treated for angina. So this is all in this session on cellular signaling. If you want to go for more, then you need to go for the plus subscription on Unacademy. So if you have any doubts, if you have any queries, then you can post your doubts and queries, and I am here to answer them. एंड इसके साथ ही साथ मैं आप लोग को बताना चाहूँगा कि गाइज प्लीज़ लाइक शेयर एंड सब्सक्राइब टू दिस चैनल as much as possible, and please share as much as possible with those who are preparing for this examination. And you can use my referral code HY10 for getting 10% off on the next Unacademy subscription. So if you have any doubts or if you have any queries, then you can post your doubts and comments in the comment box. एंड आज ही से ही हम लोग ने लक्ष्य 2021 लक्ष्य JRF 2021 स्टार्ट किया है. सो वेयर वी विल बी टीचिंग यू फॉर द अपकमिंग CSIR एग्जामिनेशन. So this is about Laksh JRF for 2021. So it has been started from 6 December 11 AM. So हम लोग की classes होंगी कल से. So myself Himanshu and Kumkum ma'am will be teaching for life sciences. Rahul sir and Amit sir will be teaching for mathematical sciences, and Richa ma'am will be teaching for chemical sciences. So आप लोग इस classes में regular रहिए. So you should be regular in this classes. So once you are regular in this classes, so it will be very useful for you for clearing the CSIR NET JRF examination, and we are here to help you. So this is all what we studied in cellular signaling. So I hope this session was useful. So यहाँ पे कई important concept discuss किए हैं हमने. So if you have any doubt, any query, you can type it in the comment box. If you want that something more should be added, then also you comment in the comment box. And guys, please like, share and subscribe to this channel as much as possible. So I am waiting for your doubts and queries. In case you have any doubts and queries, you can type it in the comment box. आप लोग से मैं जानना चाहूँगा कि आप लोग कौन सा मॉड्यूल स्टार्ट कराना चाहते हैं, नेक्स्ट कौन सा मॉड्यूल मैं स्टार्ट करूँ. सो उसको भी आप लोग चाहें तो कमेंट बॉक्स में टाइप कर सकते हैं. एंड गाइस प्लीज़ बी इंटरेक्टिव एंड प्लीज़ बी थॉरो विद द क्लासेस, बिकॉज इफ़ यू आर नॉट प्रेजेंट इन द क्लासेस देन रिकॉर्डेड क्लासेस में उतना एडवांटेज नहीं रहता है. सो ट्राई टू अटेंड द लाइव क्लासेस एज मच एज पॉसिबल, बिकॉज इट विल बी यूजफुल फॉर यू ऑल. So I hope there is no doubt or queries. So thank you guys. I'm closing off the session.